Hello, everyone. Hello, Rasmus.
Hello.
So let's wait maybe another minute and then we can start. All right, so before we start, there is a small announcement that we have the change in the time of the next lecture on October 20. It will be at 7:15 p.m. Yes, and yes, today we have Professor Rasmus Nielsen from University of California in Berkeley. And Rasmus, you are very welcome to teach us. Thank you.
Thank you. All right. So last year, oh, I can't share screen. If you can.
Yeah, just a second.
So last year, I gave several sort of overview lectures over various areas of population genetic theory. And I thought that this year I would go a little bit more in depth with some topics relating to population genetics. And much of that will focus on my own research, in part because that's what I know the most about, but it's also more general. The topic for today is models and inference of admixture and integration, which is a very hot topic right now in population genetics. And in fact, I'm working together with some project that I'll talk a little bit about now, also together with Vladimir and people in his group. So much of the work on inference in population genetics really starts in around 2000, when Jonathan Pritchard in college makes the famous paper on the method structure. And what they want to do is they want to make a model, a statistical model, where they can fractionally assign people to different populations. So the results of those kind of analysis is what you see up here in the top of the figure. Each column here is an individual. So here there are a few hundred individuals, and each color is the membership of that individual to a particular population group. So here they've been put on a map. There's different, this is Southeast Asia. These are Han Chinese, that's a major ethnic group in China, a group in India, some in Indonesia, and so on. And you can see these different, most of the individuals, they belong mostly to one of these groups. So for example, the dark blue here, those are people in Papua New Guinea. And the light blue, those are people, Yorubans from Western Africa. And so you can see, so some individuals are fully assigned to a population. So for example, somebody is fully orange here. That means that they are fully assigned as 100% from this group here. So that's a reference European population. Some Acadians, these are some people from Scotland that have been used in some studies as a European reference population. But you can also see that some individuals, they sort of fit better at being half something and half something else. This is people that fit somewhere in between Acadians and the Han Chinese. So these might be people in Central Asia that better fits, as you can better model statistically as half Acadian and half Han as fully one or the other. Okay, so this is what's known as the structure model, or sometimes admixture model. And it's been used in thousands of population genetic studies, and it's sort of a basic model that's used to assign people to different population reference samples and assign them fractionally to it. And of course, the way it's often is interpreted is that individuals, for example, these individuals here, they are at mix between the pink group and the orange group. So they would mix between Europeans and East Asians. But really, of course, they may not have any admixture in the sense that the production of recent intermarriages or interbreeding between people from Western Europe and Eastern Asia, and they may not just simply on the continuum of human genetic variation fit somewhere in the middle between these groups. Okay, so there are lots of discussion about how to interpret these plots. It's a very generic statistical model. Okay, so this is the model here. This would be Pritchard et al. 2000. That's the original, the people that first proposed to use this type of model. And the likelihood function, they have a likelihood function they define that's very simple. Hold on, I need to get things from all these things from Zoom. See if I can, that are covering my screen or my slides, see if I can get them to go away somehow. It's a little better. All right, so this is a basic model. You can think of this Gij as an indicator variable for if you sample a single gene copy in an individual, I in position J of the genome, whether it has the derived or the ancestral allele in that position, if you just look at one of the gene copies. And then the likelihood, you calculate that as the probability of the probability of the allele in that locus in the particular population, multiplied by the probability that the individual belongs to that population, summed over all populations. And then you take the logarithm of that because it's a log likelihood, and then you sum over all individuals and over all positions of the genomes. It's similar to taking you take the log sum, so that's similar to taking obviously similar to taking the product over all of these probabilities. And this is just the likelihood. If you have the other allele, then it's 1 minus the allele frequency instead. Because this Gij, you can think of that as an indicator variable. It takes on the value either 1 or 0. So you'll either have this term or you would have this term in the likelihood function. So it's a very similar likelihood function. It's a type of a mixture model with a mixture over these probabilities depending on which population you belong to. And then you have this product over all of these observations for all individuals for everywhere in the genome. Now, the implicit assumption here is that all positions in the genome are independent of each other. And also, if you're diploid, like humans are, we have 2 copies of our DNA, one from our mother and one from our father, then these 2 copies are independent of each other. That's the implicit assumption of this model. So one way to think about that is that it assumes Hardy-Weinberg equilibrium is one of the assumptions, because that's the same as assuming independence among the 2 alleles you have. So that also means that you assume that the composition of your parents, for example, are exactly the same, and of their parents, and so on. And it also does, there are also other consequences of assuming this model. There's a lot of interesting biology you're sweeping under the carpet by doing this, because there's a lot of information in getting in the pattern of admixture in the genome. We'll talk a lot about that, how the pattern of admixture, that is the spatial pattern along the length of the genome. There's a lot of information in that about how this admixture came about. This model is a model, it's a very generic model. It doesn't make any particular assumptions or trying to infer how these populations were formed. But there is, in fact, information in the data about that. So you can extend this to genotypes, and that's an equation down here where you have, instead of having just one allele, you have 2 alleles, you have genotypes, And then you can use genotype likelihoods that I know Torfinn has been talking a lot about, using genotype likelihoods. And you can use genotype likelihoods also in these kind of methods. And one method that we have developed is using these type of genotype likelihoods instead of assuming that you have observed all the genotypes with no uncertainty. It models the uncertainty in the genotype calling when you're doing these kind of inferences that you trivially can put into these kind of models. That's very easy. Like in most other things we're doing, it's quite easy to take standard methods and then extend them to use genotype likelihoods to model the uncertainty in the genotype calling. The fact that the data is noisy is modeled this way. All right, so maybe I won't talk so much about that, but we can then optimize this likelihood function. We're doing it using the sequential quadratic programming algorithms, where we're optimizing this likelihood function. So the parameters we are estimating is for each individual, the individual's fractional membership to all of these populations. At the same time, we estimate the allele frequencies in however many SNPs we have. Typically, people don't use all of the SNPs in the genome, but just a few 100,000, because there's plenty of information, just a few 100,000. So you might be estimating if we have 1,000 individuals and say 10 populations, then there are about 10,000 population parameters we estimate, plus we might estimate 200,000-300,000 allele frequencies. So we are estimating several 100,000 parameters, and that's possible because they're conditionally independent of each other. So it's actually not that hard an optimization problem. The original structure program proposed by Jonathan Pritchard et al., that was using MCMC. It makes a much harder problem, and there are lots of problems with convergence, but more modern versions, including ours, and methods implemented, for example, in admixture, a program of the name admixture. They are based on maximum likelihood optimization, and generally it's not really a problem to optimize the likelihood function. Here's a bit of here's a graph that shows as a function of the execution time in seconds for data sets with 118 individuals and 17,000 markers. The execution time of in blue admixture, that might be the standard program in this area, and then our program that I just described before with the quadratic programming, which we call OHANA. And this is the likelihood value you in multiple replicates that you get. You can see that the likelihood value we find tend to be larger. So there's, the fact that you don't get the same likelihood here tells you that there are optimization problems. But the estimates, when you look at them, they're actually very similar. So if you take a run here from admixture and one from our method, even though we tend to get higher likelihood, the estimates are actually not very different from each other. Just because there's so much data that a likelihood difference of 1,000, that's a very important, statistically important difference in likelihood, actually doesn't produce very different estimates. So despite still some problems maybe with optimization on standard methods, these methods are widely used and trusted to give reliable results. But the question is, so that's, so in the end, you can end with producing something like this. That's from our program. And you can then also, now you have the allele frequencies, you can estimate, for example, trees. How do these populations relate to each other? So you can use various analysis of this data jointly with an estimation of allele frequencies. So you can estimate, well, these hypothetical admixture groups, hypothetical pure populations here. What is the genetic relationship between those? And you can elucidate that. So for example, for this tree, you see the longest branch here is out to the Eurubans. And then there's some group here that has Europeans and South Asians. So that's people from India. And then the East Asians and Southeast Asians, they fall in these groups out here. So you can, these models are tremendously useful for a lot of different things and have been at the core of much of population genetic research on genomic data. All right, but this model, as I said, makes some very simple assumptions about everything that's going on in the genome between the two gene copies you have from your father and from your mother. And no matter where you are in the genome, everything is exactly the same and can be modeled with this very generic model. And it's in some sense not a good population genetic model. We know that that's not true. We know that lots about the way admixture is formed does not generate data. That is IID. That is where the observations of gene copies are identical and independently distributed. That kind of data does not result from this process. So in some sense, the basic model we use is all wrong. It's very, very useful, but it's not right. And the question is, what can we gain? What can we learn from using more complicated models that actually models the admixture or integration? Okay, I'll talk, the rest of this lecture will be about that topic, about modeling in more detail admixture and integration. And as you say, this word integration, biologists are using a lot these days. That means transfer of genetic material between species. So here we have two species, and there's transfer of genetic material between them. So if you have one individual going into population 2 from population 1, there's some transfer of genetic material, and eventually after some generations, some individuals will carry partly some of the material from species 1. So that's just like what I described as mixture. So the words integration and admixture, they really mean the same thing. But integration is the word that biologists use mostly when it's between different species. Individuals from different species exchange genetic material. And admixture is when it's from different populations within the same species. But if you think about it mathematically, admixture and integration is the same, and the models we use for it are really the same. All right, before I go on, I think I will stop now for a second, just ask if there are any questions right now.
Well, it's quite clear to now.
Okay, good. Please interrupt me if you have any questions about anything. Then please interrupt, and then we can discuss. I think there's a few people in the class that it's okay to have a discussion. So, and if there's something you don't understand, likely other people don't understand it either. So I will encourage you, if you don't mind, please interrupt me. That'll be better than me just speaking for 2 hours.
We will most certainly will.
Okay, wonderful. All right, so what we're going to go through now, I'll go more in detail with, is this admixture process here, how that happens, and the details of that admixture process, and thinking a little bit about that. So you can think about now an individual that is an F1, that is the first generation hybrid between two species, or the first offspring between two individuals from two different populations, that then has one chromosome from one population, the red one, and another chromosome from the other population, the blue one. And so this is in the first generation after an initial admixture event. So now we're going to think about it as admixture happened in one generation, where individuals come from one population into another population. And then we're going to look at what happens now. So after one generation, if this individual, the hybrid individual, then keeps, or if that breeds with an individual from the red population, you might have an offspring like this, where because of recombination, this offspring now doesn't have this whole blue chromosome, but just half of it, because a recombination event happened here between the red and the blue chromosome. So you might, in the next generation, have something like this, where an individual has a chromosome that's half red and half blue. After 5 generations, there might be some segments that we call admixture tracts. There's this word up here, these admixture tracts that are shorter yet. So every generation, there's an opportunity for recombination to break down these chromosomes into smaller and smaller chunks. So after 100 generations, you might have individuals in the population that have lots of small little trunks on different chromosomes, like this, these admixture tracts. And what we can use that for is we can date when that admixture event happened. So in evolutionary biology, one of the core tools that's being used all the time is what's called the molecular clock, that evolutionary biologists are very used to. Use them when you date anything in evolutionary biology. You take phylogenies, you want to figure out when have species diverged for each other, you use a molecular clock. That's a clock where the units that you're counting are mutations. You count how many mutations there are between different individuals or between different species. And you can then use that to infer, if you know the mutation rate, how long time since those species diverge from each other. But here we have a different way of dating events. We have a recombination clock. We can use the rate at which recombination happens to date admixture events, because the length of these tracts determines, if we know the recombination event, can be used to estimate when the initial admixture event happened. And so that's what's going into much of current research on trying to date admixture events, trying to get at the length of these tracks in one way or another way, directly or indirectly. It's also the core of much of the sort of commercial human genomics. So, in the United States, there are all these companies where you can send your DNA to them, and then they will do all kind of analysis of your DNA for you. For example, 23andMe or Ancestry.com. They're very large companies, and there are millions and millions of people that have sent the DNA to these companies. And one of the things that they deliver, and this is from 23andMe's webpage, I took that a couple of years ago from the webpage, the results is sent for an African-American man that has sent in his DNA to them. You can say you get these are the chromosomes, these admixture tracks here. And you can see something is painted as being either European or African and Asian in this individual. And there's these relatively long tracks that are either African and European that represent admixture after African people were brought over as slaves to America. Then the admixture that happened particularly in the first generation between Europeans, people of European and people of African ancestry. So you can see all these tracks in the genome. So 23andMe, part of what they're doing is that they're painting the genome and doing this what's called a chromosome painting, where they paint the genome according to ancestry. You see, this individual apparently also has 2% Asian. And what you always wonder when you see something like this, does this individual really have 2% Asian DNA? Or is this some kind of artifact? For example, of the reference populations that are being used are not quite accurate. So it's sometimes easier to paint something as being Asian than, well, that gives the best statistical fit. But this is what you get if you're if you send your DNA to Ancestry or 23andMe or Ancestry.com. And I often get emails myself with people that say, for example, oh, it says that I'm 0.5% Japanese. Can you help me interpret this? And of course, typically people that have these half percentages, it's some spurious results that comes out. So there's a lot of interest in these methods, in part because they have all these commercial interest, because people actually pay a lot of money and companies are making a lot of money on making this kind of ancestry inference, because it's a lot of interest to people. What is their ancestry? So that's another interest in these ancestry tracks, the way they're being used. So to my knowledge, the first time anybody actually used ancestry tracks to try to estimate divergence time or admixture times between populations is some work that I did about 15 years ago together with my postdoc back then, John Poole. And that was not humans we were looking at. We were looking instead at mice. So in the house mice, Mus musculus, there's this famous introgression zone that population geneticists have been interested in that goes right through Europe. In Eastern Europe, in Russia, you have the Mus musculus subspecies. In Western Europe, in Spain and France and England, they have Mus musculus domesticus. This is the one that the common house mouse, and this is the one that the lab mouse has been domesticated from. But they're quite genetically differentiated from each other. And many questions has been on when did they come into contact with each other, and how long time has there been this sort of hybridization zone there between the two subspecies? They can interbreed, even though they're quite genetically differentiated from each other. And what we did, took some SNP genotyping data, and then using relatively simple methods,
It seems to be some kind of problem. Is it from my side or for everyone else?
everyone else.
Okay. So I guess we're just waiting for Rasmus to come back to us.
So I'm really sorry about that. I lost internet connection. What was the last thing you heard from me?
The mice.
How far did I get with the mice? Did I talk about, let me share my screen again.
Sorry?
Did I show this slide here?
No,
No, you didn't.
Did I show this slide?
Yes.
but you didn't tell much about it.
Okay. So sorry about that. I will, if this happens again that I lose connection, I will try to log in from my phone with the cellular network and try with that instead. But the lecture might be a little funny if I try to do it that way. Let's hope that I don't lose internet connection again. Anyway, I was at these mice and talking about these introgression zones between the mice and the fact that you could go in the mice and then look at these tracks, and these mice subspecies are so genetically differentiated from each other. It's really easy to go in and find these tracks. They were relatively long and very clearly differentiated, much more so than in humans. Humans, it's much harder to find these tracks, but in mice, it's very easy. In these two subspecies of mice, it's much easier because they're very genetically differentiated from each other. Then when we can go in, so now we have a distribution of these track lengths in mice. And now we can fit different models to it. And so the way we derived the models in this paper, so this is back from 2009, it was published. This is research we did in 2007 and 2008. And long time ago. And the simple model we assumed here is that basically the model similar to what you saw before with the admixture tracks. And we just assumed for simplicity that these admixture tracks, they don't interact with each other. So they don't recombine with each other. And they're sort of just being broken up by, can you hear me?
Yes.
Yeah, sure, we do.
Okay, good. So they're just being broken up with by recombination, and then they're sort of floating around randomly in the population. And then you can it's very easy to make models then of what's the distribution of these tracks, because recombination events are laid down according to a Poisson process. The waiting time is exponentially distributed. And so the waiting time, the length distribution is just an exponential that depends on the age of the tract. And what you can do is you can take basically for any model that you have and integrating over the time in which there was migration from one population to another, and then gets relatively simple expression of the length distribution. And so this is for a model in which migration is constant. Here in blue, here we have always had migration between the two subspecies. In red, that's one where there's been migration in the last 100 generations. In green, the last 200, and in yellow, the last 300 generations. So
May I have a small question, please?
Sure.
Yeah. Maybe I missed something. I should understand how it would work if we would have two absolutely different genomes. But if I assume that the genome of one type of mice and another do interlap a lot, and well, does all this assumptions hold if we know that the genomes may be different in significantly smaller number of places than the whole length?
Yeah, no, I think it's a good question. Does it even make sense to talk about if, you know, that
Yeah,
you can
sure.
find mixture tracks when you always had a mixture between them? Is it, you know, is that a good abstraction? And I would say maybe not. I mean, in reality, you probably could find some segments that was more one than another. But if there really has been constant migration like this, probably admixture tracks is not a good way of thinking about it, except maybe if it's very, very little migration. So they're still highly differentiated, and you just find sort of these few tracks. But at some point, there would be some older tracks you then can't really infer and so on. There'll definitely be some inference problems that really old tracks that probably are really, really short. You can't really infer them easily, right? Because so I think it's not a good model, no. So the short answer, no, it's not a good model.
Okay.
But no, with that abstraction, you can still make these calculations and see in that limit what would you see. And then you can, you know, these models maybe make more sense where it's only been recently you had a mixture, and then you see, you know, some figures, some graphs more like this. So it's clear that there's information about when, if we assume that's been constant migration between the two populations, this distribution of the track links provide us with the kind of information that we are interested in, and we can then fit models to that. And this is the best fitting model that we got here. We tried different models. The best fitting model we got was that there was a change in integration rate approximately 200 generations ago. So that 200 generations ago, we got there's much more integration between the two subspecies. That's where it started. We can also do like a ratio test and so on, test hypothesis about that. And the story that we wrote, this fits with when you started to ship wheat around in Europe. So before that, you would have gotten all your wheat locally. But at some point, you started shipping and also getting railroads where you were sending wheat back and forth in Europe between different locations. And the mice traveled with that. And that's how mice have sort of been spreading in Europe. And so it makes a lot of sense that you started having a lot of integration between these subspecies about 200 generations ago. Okay, so that was the sort of story that we told there about that and the estimates that we got about that. Let me see here. So the next, so then for a little while, I didn't think much more about this. There wasn't so much data to work on this, in particular in humans. It's much harder to find these admixture tracks. They're not mice. Humans, we're much more closely related to each other, and it requires more sophisticated methods to get at these admixture tracks. And back then, we didn't have those methods yet. We do have them now, but we didn't have back then. But then I got involved in another project that was looking at the Neanderthal DNA with Svante Pääbo. And the idea back then was that Svante was sequencing the very first Neanderthal genome. And we had developed methods for trying to find integration among in humans. And then we worked with Svante on finding that, showing that there was integration between human and Neanderthals. As you probably all know, we know now that there is, in fact, has been integration with humans and Neanderthals. And so we got started working on this with integration again, just to try to understand this integration between Neanderthals and humans. But then as we worked on other projects, we also kept stumbling into integration. There seemed to be integration everywhere. And I'm going to go into sort of the biology and give you a few examples. Something, and I think I mentioned that before, that we worked a lot on, is this Tibetan altitude adaptation and trying to understand that. So if you go up in the height of the Tibetan plateau, it's in an altitude of 4 kilometers. So in that altitude, the partial pressure of oxygen is only 60% of what it is at sea level. So with each breath of air, you only get 60% as much oxygen as you do at sea level. So so you'll suffer from oxygen deprivation. We would all do that if we go up and stay in a longer time at the Tibetan plateau. There's then a physiological response to that. Our body reacts to the fact that it doesn't get enough oxygen. It starts to increase more red blood cells. Remember, red blood cells, that's what transports the oxygen around in the blood. So if we have more red blood cells, we can transport oxygen better, and our lungs need to get less oxygen that way because we can utilize it better, we can transport more of it. That's a good thing, because now we get oxygen out to our to our vital organs and everywhere in our body better. But there's also a negative effect of that. It increases the blood viscosity. The blood becomes thicker. Instead of having nice, clean running blood in your veins, you now have something as thick as yogurt that's pumping through your veins, and that's not good for you, because it might give you higher blood pressure. It might give you all kind of other negative fitness consequences, increased risk of stroke, and many other things. It's associated with chronic mountain sickness as well. So it's a bad thing overall for most of us. Now, what's interesting is, and that's something first described by Cynthia Beale, an anthropologist, is the Tibetans, the indigenous people in Tibet, they don't have that problem. They produce just as much red blood cells at high altitude almost as in low altitude. What you measure is hemoglobin concentration. Hemoglobin is a protein that sits outside of the red blood cells that binds the oxygen. And so if you're more hemoglobin, that's because you have more red blood cells, because you can't pack more hemoglobin on the red blood cells. They're already fully packed with it. So more hemoglobin means more red blood cells. And what she showed is Tibetans, presumably as an adaptation to high altitude, they actually don't overproduce red blood cells as the rest of us do in high altitude. They don't have this increased viscosity of the blood. Their blood is not too thick. They have other ways of coping with a high altitude. So what we did back then is we did a long time ago, 10 years ago, we did a scan in the human genome for selection. And I talked a bit about these kind of methods for scanning for selection last year. And many of you are in some degree familiar with these methods. We looked at the genome to try to figure out where there positions in the genome can we find individual SNPs that are highly differentiated in Tibetans, particularly in Tibetans relative to other populations. And there were two genes that stood out, in particular one, EPAS1, that was really highly differentiated in the Tibetans. That means that there was an allele frequency that was very different specifically in Tibetans compared to all other populations we compared to. All right, so what does EPAS1 do? Well, it's what's called a transcription factor, which regulates the response to hypoxia. So it's a gene that regulates how other genes should behave. And it's particularly acts in response to low oxygen. So hypoxia, that's low oxygen levels. So when that gene gets activated by hypoxia, by low oxygen, that we know that regulates red blood cell production. And in fact, what we could go in and do is we could do a little association mapping study on where we compared EPAS1 genotypes in different Tibetans and looked at their hemoglobin concentration. Remember, that's a proxy for red blood cell concentration. And we could show that the type that seems to have been under selection, the genotype, if you're homozygous for that, you had much lower hemoglobin concentration and therefore also lower red blood cell concentration if you had that. So we found the genetic variants that indirectly or directly is responsible for the altitude adaptation in Tibetans, where they don't overproduce red blood cells. So that was interesting. We had narrowed down to this gene EPAS1. It doesn't have to be this particular genetic variant because of linkage disequilibrium. It could be many other genetic variants that are sitting in the same region, but it certainly locates to this particular gene. And there are other studies using expression data and other data that suggest, well, in fact, this gene is the causal gene. So, of course, what we wanted to do now is we wanted to try to understand how selection has been acting on this gene. Okay, so we wanted to do more detailed population genetic modeling of when selection started to happen, how strong the selection is, and so on. And the problem that we had was that none of the models of natural selection that we usually can fit to the data seem to fit to the data here. Okay, and I'll explain to you here with this figure why it didn't fit the data. So these are individuals that are Han Chinese, against that's a major ethnic group in China, and Tibetans. And each column here is a SNP. And you either have the derived type, then you're black, or the ancestral type, and then you're white. So each row here is a haplotype from either Han Chinese or Tibetans. And what you can see here, just looking at it, is that these the two haplotypes that are highly differentiated from each other. Okay, this haplotype up here, that's the common one in Tibetans. That's the one that carried the genetic variants that are associated with decreased red blood cell production in high altitude. And this haplotype is very different from this haplotype down here. All right, and this actually so differentiated, it's not explained by simple models of natural selection. So the way you can think of natural selection might act is that you have a new mutation that arises that gives you a fitness advantage, and it lands on some chromosome, on some haplotype. And then that haplotype will increase in frequency in the population because it's favored by natural selection. But that in itself will not cause that haplotype to be so differentiated from other haplotypes. So this pattern of highly differentiated haplotypes, we couldn't explain it with simple selection models that we had. So we thought, maybe it's Neanderthals, maybe it's because of integration from Neanderthals. And that is a segment of Neanderthal DNA, but that turned out not to be true. But then we compared to DNA from another group called the Denisovans. And then there was almost a complete match. So the green here, that's the Denisovan sequence. And it almost completely matches this haplotype that is associated with altitude adaptation in the Tibetans. So not all of you may necessarily be familiar with the Denisovans, but when Svante Pääbo, he was sequencing more and more Neanderthal DNA. One day, some paleontologists, they gave him a little piece of a finger from the Denisovan cave in South Central Siberia. That's a place where there's lots of Neanderthal remains. It's in Russia. There's lots of Neanderthal remains in that cave. And it was a little piece of a finger bone that he got, that the anthropologist says, this kind of looked like it could be Neanderthal. And so he got to grind that up and get DNA out of it. And that turned out to be from something different, not exactly Neanderthal, but a very distant cousin of Neanderthals, something that's now called Denisovans. And we don't know them much from any physical remains. We know them from some teeth. There's a jawbone that's been found actually in Tibet that people claim based on protein sequences, not DNA sequences, might be Denisovans. And then we have some of these finger bones, but we don't really have many remains of Denisovan, physical remains of them, but we have the DNA. And in this region of the genome, in EPAS1, it almost exactly matches this Tibetan haplotype. Okay, so then, so what can we do with this? For example, can we argue now that this is in fact introgression from Denisovans? Couldn't this be just shared ancestral variation? Okay, and the arguments that go into that, when you want to make those kind of arguments, is first of most the length of the fragment. Because if the fragment is something that's really, really short, then it's unlikely to be something that's recent. But if it's really, really long, then it's likely to be recent. So we can use this recombination clock to make arguments about it. So then, and we can also use a molecular clock to make arguments about it. So we can look at the number of difference between the haplotypes and ask, okay, how many differences are there between haplotypes? So if we take the Denisovan haplotype, for example, the number of differences between that haplotype and other human haplotype is vastly larger than you would expect by any model of selection. So these are two different selection models we simulate under various different conditions, and the number of fixed differences between the Denisovan, the haplotype that we see in the Tibetans and other haplotypes is much longer than it should be. But we can also show that it's much shorter between Denisovans and the Tibetan type than it should be if it was an integration. So in addition to this length of the fragment, we can also use the number of mutations. So one way to look at and summarize the genetic variation in the region is through this kind of plot here that people sometimes make a haplotype network, where they take each pie chart here as a haplotype, and then you get connected to the nearest, to the closest other haplotype that's closest to you in terms of number of differences. And the coloration of the pie chart here corresponds then to different populations. So for example, This is Han Chinese individuals. The red here are some Europeans. This purple is a Finnish population and so on. And the pink here, those are Tibetans. These are other populations, other human populations that are the closest to the Tibetans in the region. And these are Denisovans. So there are 9 differences in this region between Tibetans and Denisovans. There are 40 differences between Tibetans and other humans. And there are 35 differences between Denisovans and other humans. So not only are Tibetans much closer to the Denisovans than they are to other humans, but in fact, Denisovans are closer to other humans than Tibetans are. And that's sure. Let me just finish this sentence and then I'll let you out. So I'll just explain why this is. So the reason why you find that is that this is ancient DNA. That's about 50,000 years old. So that there's been 50,000 years more to accumulate differences between the Tibetan haplotype and the human haplotype than there has between the Denisovan and the human haplotype. And for that reason, Tibetans are even further away from other humans than the Denisovans are, because this is ancient DNA. All right, please, with giving the question.
Yeah, the question is the following. As far as I understand, there is extremely small amount of genetic material from Denisovans, and of course, it is ancient. How sure can we be that our results are like, well, accurate, and we do not deal with some, maybe a random variation or something, just how reproducible it is?
I mean, it's highly reproducible.
Can we be sure?
Yes. I mean, it is reproducible in the sense that we do have Denisovan genomes that are relatively high coverage, and several of them. And so you get the same
several
thing.
of them. Okay, good.
You get the same thing in several Denisovan genomes. So by now we have plenty of Denisovan DNA that's quite high coverage.
Okay,
so,
thank you.
I mean, it's also, another thing is what's the chance of introducing errors that by chance make great similarity? So you have to put in let's say it was a contamination by a human and errors. You have to put in 31 errors in just exactly the right spots, where you revert from a difference to a similarity and don't introduce any other errors. So creating similarity by errors is very difficult. Creating differences by errors is easy, but creating false similarity by errors is very difficult, I would say.
Yeah, we should have been extremely unlucky to do that.
Yeah, I mean, you could make sort of calculation of, and get 10 to the minus 48 or something of hitting exactly those positions and so on. So I think, I mean, the worry is always, well, what if it's contamination and so on, right, with ancient DNA? I think that's sort of a major worry. And then, of course, if there's something you see is differentiated, well, is the data of so low quality that you have higher divergence because of errors in the sequencing? That would be the main concerns, I think, normally. But it is good for ancient DNA to think about the error structure and take that into account. And I think Thorfinn has already talked with you about that, right? Or did they give the lecture? Or somebody gave a lecture on that, isn't that right? Or we'll do it at least. All right, so I'll give, let's see how I'm doing on time. This is 2 hours, right? I have 2 hours here. Is that correct?
Vladimir, I guess
yes, 2 hours.
2 hours, okay. All right, so I'll give another example of some work we did where we also ran into integration, where again, we found integration. This is a study we did on Inuit from Greenland, where we were working with health authorities in Greenland and trying to understand more about the genetic background of the Inuit, the indigenous people that live in Greenland, and worked on type 2 diabetes and other things. But what we also did was a selection scan. So we go around along in the genome and try to find positions where the Inuits are highly differentiated from other populations. It's the same thing as we did in Tibetans. Now we did it in the Inuit. And our top genes were these FATS genes. That's a whole interesting story in itself that I'm not going to talk about today. But the second peak that we found was this peak, TBX15 Wars 2. And for these ones, these genes, we know very well what's going on there and understand the physiology. There's a whole story behind that. This one we don't quite understand, but we have an idea about it. TBX15 is involved in differentiation of adipose cells and adipocytes. These are the fat cells. And roughly speaking, humans have two types of fat cells. Bad white fat, that's all the fat as you get older, like me, that starts accumulating around your waist. You don't want to have too much of that. That's unhealthy for you to have too much fat. But there's also the good brown fat. So that's brown. These are fat cells, adipocytes, that are brown because they contain many mitochondria. Remember the mitochondria, they're the powerhouse of the cell. That's where energy is being converted. And that's also where heat is being produced. These are essentially heat-producing cells. They burn calories and produce heat. Babies have a lot of them when they're newborn. That's why babies can keep warm, because they have, as long as they're being fed well, because they have a lot of brown fat. As adults, we have very little on it. 20 years ago, we thought essentially nothing, but now we know there's a lot of brown fat, particularly on the back, but also other places that's interspersed among the white fat. And there have been anthropologists that have been claiming that there's actually more brown fat in the Inuits. We don't know that for sure, actually. They're not really good controlled studies of that. But that's a claim that's been out there. And this gene, what TBX15 does, is that it's associated with determining whether a cell should, an adipocyte, an early one, should become a brown fat cell or should become a white fat cell. So the hypothesis here that we don't have evidence for, but we have collaborators working on seeing if they can show this, is that this is in fact has to do with adaptation to cold climates, and that the increased production of brown fat cells. That's a hypothesis. We don't know that. But one thing we could see is that this gene was also integressed, also likely from Denisovans. So this is a haplotype network again for this region. And this is most human haplotypes fit in here, but then there's some other ones here, including the ones that is at 100% in the Inuits, but also exist in other populations. It's many populations in the Americas, and East Asian populations also have this haplotype. And these are the Neanderthals, and these are the Denisovans. And it's again much closer to the Denisovan haplotype. And again, by arguments of the length of the haplotype and molecular clock arguments about the number of differences between these haplotypes, we can show that this must also have been integration. That this is something that this divergence here is something that must have happened within the last 100,000 years, and the divergence time between humans and Denisovans is 500,000–600,000 years. So this is another integration event where, again, as humans adapted to new environments, adapted to those environments by picking up genes by integration with other individuals that likely already lived in those environments. And so part of the story about human adaptation to new environments is that humans actually get these genetic variants done it historically 10, 20, 30, 40, 50,000 years ago from other hominids that live in the environments, we think. And there are more and more evidence that this is in fact the case, that as humans spread out of Africa and encountered these radically new environments like high altitude in Tibet and extreme cold in the Arctic, there were other humans that were already near those environments, and humans could interbreed with them. Modern humans could interbreed with them and get genes from them that are adaptive in those environments. And many of the genes that we have that are associated with adaptation, for example, to cold or to high altitude, we got them from other hominids like Neanderthals and Denisovans, and perhaps other species as well that are yet to be characterized. Okay, so that is enough of these examples, but these are just some examples to also show you why is there so much interest about this, how integration works today. It affects human health and many other things to try to understand these processes. So this one here, this old project I talked about with the mice, we assumed independence with all these track links, all the tracks, that they're sort of segregating around in the genome independently of each other. We can make models of that. That's mathematically very easy. It's very convenient to do that, but it's not right. Of course, they can recombine it also. And so since we started doing some other projects, we would start to look more into what is the actual distribution of track links in the expected in a genome. And these are some simulations and some analytical results for some models we developed. And these, in each of these plots, there's the different migration rates. Here you have m equal 0.1, m equal 0.5, and m equal 0.9. And then you have different times since admixture, 5 generations ago, 100 generations ago, and 2000 generations ago. And then we have analytical results for some models, and also simulations from a Wright-Fisher model, which we consider the truth. So that is the solid black one here. So you can figure this. This is what the what the admixture track distribution actually will look if you take sort of the full complexities into account. Now, some models are not, when at longer genetic distances and at older admixture times, they become very inaccurate. This, for example, from the SMC model and a previous method that's been used to model track lengths. These models, they don't model the correlation structure well. So you don't get really good approximations to this distribution of track lengths. You also have something, well, best model SMC prime still doesn't really do very well at modeling the distribution of track lengths, and it's only really at best an approximation. The full red coalescence model, that is sort of our standard model, the coalescence with recombination, does a good job at modeling integration track lengths and their distribution. But several of the other models that we commonly use, they're not really good at modeling sort of long distance correlation structure, and that creates a problem for some of these models. It's also when admixture time is large. When it's short, all of the models, even the coalescent model, has problems. Okay, so why is the coalescent model inaccurate in those cases? Well, that's because in over a very short amount of generation, there's some complexity of pedigree structure that is not modeled by the coalescent. So the coalescent model is an approximation for large population sizes, and essentially looked when you observe over many generations. But over a short amount of generations, the fact that truly in diploid population you have this more complicated pedigree structure actually make the coalescent also inaccurate. So the true structure of these admixture tracks is actually quite complicated to fully model if you want all the complexities there, and are not even modeled well by the full coalescence for ancestral recombination in an ancestral recombination graph. Because many of the models do pretty well in many conditions. So, for example, under intermediate amount of admixture time, most of these models are pretty good. And what you can see is that you get sort of what we assumed for these mice, an exponential distribution. This is on a log scale, so this is an exponential distribution of the track lengths. That's pretty good. But if divergence time is too large, there's effects of genetic drift and back recombination that make many of the models inaccurate. And for short amount of admixture time, models are also inaccurate because they don't model these complexities that come from the pedigree structure, right? Fortunately, for many applications, we are in this, sort of more along this type of scenario. And even for longer migration rate times, we can use certainly the SMC prime or the coalescence to model low level of migration. The reason why it's different for high level of migration is the migration tracks then recombine with each other much more. And that's what creates these complexities here. That if you then don't model the correlation structure well, then there's a problem. Okay, so that's just a little bit about the complexities of this. All right, so some of the observations here, deviations from macro process occur both when time is very recent due to pedigree structure and when time is very old due to genetic drift and back recombination. So what I mean by macro structure is that these exponential distribution, when you have exponential waiting times, that's what the prediction if there's a macro process running along the length of the genome. And that's essentially what you assume in many of these methods, that you can use a hidden macro model to do the decoding, to assign these tracks. But that's actually not true always. But under some scenarios, it's true. But for example, if the pedigree structure, it's not true, or if divergence times are large, it's not true anymore. Okay. The other thing is that comes from this, and what causes some of these problems here is that admixture tracks tend to be clustered in the genome. They're not randomly distributed in the genome. And that's something that's been much of the literature has actually caused some problems. I think some wrong inferences, people weren't aware of that. Now, why are they clustered? Well, this is because these clusters represent haplotypes that were introduced early on, that are relatively large, tracks that are large, and then are being broken up to smaller and smaller tracks. That process of having large tracks that by recombination are being broken up to shorter tracks creates this kind of clustering of tracks in the genome. Why is that a problem? Well, it's a problem because this clustering in the genome, people have interpreted as being functionally important due to natural selection and so on. That, oh, if there's a cluster here, there must be some important biological reason for it. But in fact, you would expect that to happen randomly due to the way that their mixture process works. Okay. All right. So we can do some more modeling of this. Let me see. I'm missing a slide I wanted to show here. All right. Unfortunately, I'm missing a slide here. But a common way to model admixture times is by looking at linkage disequilibrium. Okay, so if you want to estimate, well, maybe before I go on to that, let me just hear, are there any questions to any of this right now? All right, no questions to this. Don't forget to interrupt me if you have questions. Okay, so a common way of trying to estimate admixture time is to look at what is the linkage disequilibrium in the genome between markers as it's affected by admixture. Admixture will create increased linkage disequilibrium. For example, if you look at a region here, there will be linkage disequilibrium between markers here that are both blue because they came in from the same tract here. So if you look at this position in the genome and this position in the genome, there will be increased linkage disequilibrium. And that's often used, this increase in linkage disequilibrium due to admixture, that is often used to estimate admixture times. And I think maybe the most commonly ways of estimating when admixture happens is now just simply looking at, or not simply, but using various methods that look at the linkage disequilibrium and the way it decays with distance, and use that to estimate admixture times. So classical linkage disequilibrium, you can think about that as the covariance between indicator variables for alleles in two different positions in the genome, position X and position Y. So that's one way you can think of the linkage disequilibrium. What is it? It's a covariance between different positions in the genome. Now, one thing we've been working on, and this is some work that originally started by my graduate student, Mason Liang, many years ago, and we're now finishing up together with Vladimir and his student is we're looking at, his postdoc is we're looking at extending this work from classical linkage disequilibrium, where you look at two positions in the genome, to looking at three positions in the genome, because that allows us to estimate more parameters. In particular, we could estimate when several admixture events happen. So what we're doing now is we're extending the concept of two-point linkage disequilibrium to three-point linkage equilibrium. We're looking at three position X, Y, and Z. And then we're looking at the three-point covariance between these, defined as this. So similarly to the way you usually define a covariance, this is the three-point covariance defined as the expectation of the value for each point minus its expected value, and then the product over the 3 points, and then the expectation of that product. So that's a natural three-point extension of covariance, and or similarly a natural way to extend the classical two-point linkage disequilibrium to a three-point linkage disequilibrium. Now, what we need to get is to be able to get at at this term, we need to be able to calculate, well, what's the probability that if you look at 3 different positions in the genome, position X, Y, and Z, that they are all admixed. So equal to 1 could indicate that they're admixed here and indicate a variable for that. Or what's the probability, the Y and Z, the joint probability that they're admixed, and then multiplied by the probability that you're admixed in position X, and so on. And the product of independent probabilities or marginal probabilities of being admixed in position X, Y, and Z. So if we can calculate these probabilities, then it turns out, then we can calculate what we would expect this covariance to be, what the covariance is. Now this matrix here, if we write this in matrix form, these probabilities, we can write that as a product of these, of other matrices that represent genetic drift and recombination and migration. So there's a matrix, the 3 matrices here. This is this 3 matrix in one generation, generation I, and the previous generation I-1. And you can iterate to get these probabilities by multiplying these matrices on that matrix. And these matrices, there's a diagonal matrix here that has migrations in each generation in it. So there's a migration term, so migration terms here. Then there's a matrix that has to do with genetic drift and the way genetic drift works. And then there's another matrix that has to do with recombination and how recombination works. So we can this way model the effect on generation I-1 of migration, recombination, or drift on this matrix of these probabilities. And you can then, based on that, derive expression for this 3-point covariance. So for example, the expected 3-point covariance here with a single migration pulse, we can write that as this. We can derive that's what that is. And then we can do it more complicated. You can do other migration scenarios. And so, for example, one thing we looked at is if you have 2 migration pulses, and then see, okay, how well could we estimate that from this 3-point covariance function? Now we can take all positions in the genome, get this 3-point covariance function, and then try to ask which model of admixture fits best. And these are just some results that show that the estimates we get with 2 different admixture times, one here, 20, 10, and 5 of one admixture, another one, 2, 5, and 10. And then these are the estimates we get when we jointly estimate for the T1 admixture here. So when the truth is 5, 10, and 20. And you can see there's a lot of noise here, but we can roughly get reasonable estimates for the admixture times. So we are able to now estimate by using not just 2-point linkage disequilibrium, but by using 3-point linkage disequilibrium, by jointly looking at 3 points, the genome and the covariance between the alleles in these different points in the genome, we can estimate admixture time of 2 admixture events. So, and we could look at other models as well, trying to get at what are in fact the times at which admixture happens. Okay, so that's one way we can use this kind of logic. There's another way, and that's still unpublished work. We have a paper on bioarchive. Go ahead.
Sorry, I have a small question. May I? Yeah?
Sure.
Yeah, sure. The question is, as always, about robustness, because the hypotheses are well defined in such a way that one hypothesis may be quite close to another one. For example, if you have just a small admixture, and as far as I know, the multiplication of matrices may enlarge the error quite a lot. So what are the boundaries for the method to be applicable? Or maybe I just missing something and...
Yeah, no, I mean, that's a good question. So the numerical issues of like
sure.
That a bit and tried, and that we actually had some problems with that, with the numerical accuracy. But I think that's sort of a practical question. Vladimir, do you want to answer that question? Vladimir can maybe better answer that than me if he's on still. I don't know.
I'm afraid I can't comment right now. And Mikhail is not here. By the way, Mikhail is an undergrad student.
Oh, it's an undergraduate student. Yeah, I was going between graduate student and postdocs. It's Mikhail. What's his last name?
Shishkin, but he's not here for some reason.
Okay, so it has been working on that and actually re-implemented that. So my graduate student Mason Liang made one program for doing this, but then he graduated and we never published it. And Mikhail has then been re-implemented it and has been working exactly on that issue about numerical accuracy and trying to get it numerically accurate. I don't know exactly how far we are with that, but it's a good point. I mean, obviously, for some model, we can write down very simple solutions like this. There shouldn't really be any problems with numerical accuracy in evaluating this. But of course, for more complicated models, we'll have to think carefully about how to do this. There are actually some tricks here, I think, so that can make it simpler, where you actually don't multiply these matrices on each other, but you diagonalize them and they have some shared left eigenvector. Some of the eigenvectors, I think, are shared between some of them. So some of this can become quite simple. But anyway, by diagonalization, I think there are things you can do that so it becomes, you don't just iterate multiplication of these matrices for many generations. They're numerically more stable ways of doing that.
So right now we can replicate Mason's results like very accurately. So
yeah. So they have replicated his results. So get the same for various times, get the same results as him. So so that's good, at least. Mm-hmm. All right, more questions about this? But the numerical accuracy is a good question because it is one of the things that we're struggling with some of these methods, stabilizing that. All right. So I'm going to talk about some other research that we have been doing. We also did together with Mason. That's also unpublished. There's a bioRxiv article on that, and I'm hoping together with Vladimir and maybe Mikhail or maybe somebody else to keep working on this next and get this out on a journal publication. And it's, to me, quite interesting. It has to do with what is the variance in admixture proportion among individuals. So remember, I was showing some plots like this before. Each column is an individual, and then you color according to the fractional membership of a hypothetical original population. So this is an example from an African-American individual that is colored according to African or European ancestry. Red would be the European ancestry and blue the African. And you can see African-Americans in the US, most of them are admixed between European and African ancestry. There are not many African-Americans that only have African ancestry, but typically mostly African ancestry. Some have almost only African ancestry, and some have it's more like 50-50. But you can see there's a lot of variance here among individuals, right? So they vary quite a lot in how much ancestry they have that's European and African. And if you imagine, that you have imagine it as a population where one population come in admixed with another population. Initially, there will be a lot of variance like this. But then as time goes on, it will homogenize. As individuals, if there's random mating, individuals will exchange chromosomes, and the admixture fragments will be broken down to shorter and shorter and shorter segments. So you would expect with time a homogenization. This is a simulation showing that here, where I simulate data from different individuals for 5 generations after admixture, 50 generations afterwards, and 500 generations afterwards. So you can see in the beginning, there's a lot of variance among individuals. After 50 generations, it's become smaller. And after 500 generations, there's very little variance among individuals within each population. Okay, so clearly the variance among individuals is also informative about the age of admixture. You can also use that to try to estimate when admixture happened. Should also make a little note, I'll get back to that you end, even though these simulations are done on the exact same conditions, you end with different levels of admixture. And that's because of genetic drift. That in these early phases, you have different amount of, some chromosomes randomly get lost. And because of that, and some other ones increase in frequency. And because of that, these different populations might end with different admixture proportions. even though they started with the same due to genetic drift. But within each population, that increasingly all individuals will have the same admixture proportion. All right, so we can get at that. What's that variance within population? And we can use, it turns out you can use the same trick as before, or similar tricks as before. The sample variance Mason showed in his thesis can be written like this. This is, again, you have an initial matrix, and then you have some matrix that operates on that with recombination and genetic drift. So here's the no migration, because we assume that migration just happened at one point, and then there's no migration after that. And then you have to do an integral along the length of the genome of this. So you have to integrate this function along the length of the genome. So you prove that this is true. That's the sample variance that you would get within a population. Now, we can then put in what's called Haldane's mapping function that assumes that there's constant recombination rate in the genome. You don't have to assume that. You can assume more complicated function, then it becomes numerically more complicated. But if you assume Haldane's mapping function, you can write down the expressions like this, that you can evaluate. This integral can be evaluated numerically quite easily. So you can write down sort of a relatively simple formula for the variance among individuals that depends on population size. That's because that's genetic drift, number of generations since the admixture happened, the initial admixture fraction, and then the length of the genome in recombination units. So that's what it depends on. And you can write down sort of this relatively simple equation for the variance among individuals. All right. So that's this here. And we can show by simulation that that is exact, that variance. Now, Verdu and Rosenberg, that's actually not it. But we can show by simulations that that's exact. But what we can also do is we can model this variance among populations here. So you can use similar tricks. So not just to get the variance among individuals, but get the variance among different populations. So we take that mixture fraction here. So that's, when I say different populations, between an individual sample here and then, you know, a random individual sample from any of these populations. So this is the exact formula that we have, that we can show by simulation is correct. And the thing is that that variance doesn't go to zero, because there's some variance, residual variance among populations here. But Verdu and Rosenberg has had a paper where they didn't model genetic drift, and then they got the variance to go to zero. So basically their model corresponds in our model to removing this term here and not having genetic drift. And of course, if there's no genetic drift, everybody ends with the same admixture fraction, and this goes to zero, the variance among individuals. So what goes to zero is the variance within a population, which they didn't derive. But the variance among individuals from different populations, that should not go to zero. That goes to something finite. So you can model that. We can model variance within population, and we can model the variance between population. And then we can use that also to estimate things about admixture times based on the variance. So this is variance within a population. We take African Americans, and then we say, okay, if we model admixture between Europeans as Americans as having, or Europeans and Africans in Americas, having started at a certain point in time, G start, that's the time at which the mixture starts, and then stopping at a certain point in time, that's G stop, then we could fit the variance among individuals to a model like that. Okay, the variance and the admixture proportion, and what we get then is a model in which admixture started 11 generations ago and then stopped after 2 generations. Okay, so that's probably, it's very simplified model. That's not, it's infinitely complex, right? With, involving millions of people, with, intermarriages and not intermarrying, in all kind of different time frames. But what this shows is that the vast majority of that mixture happened in the first 2 generations. So this fits well with the time at which slaves were brought to the Americas. And this is the first 2 generations. So this is during slavery. And what it is it's sort of the legacy, the genetic legacy of rape of slaves in the Americas. That many, rape of slaves was really, really common. And much of we call admixture is really sort of a fine word for what really happened, which was slavery and rape. That most of that mixture happened while these people were not free, but were slaves. So this is a little bit scary to think about that way. Right now in America, there's actually not that much intermarriage. So much of that mixture that we see is really a product of what happened in the first generations. All right, here's another example that I'll go through. These are the Rapa Nui. So you may not know what the Rapa Nui are, but they're the indigenous people of the Easter Islands. So Easter Islands, is this Polynesian island that's famous for having these massive statues there that are a little bit, curious who built them and so on. We know that was the original Polynesians that came to the island. It used to be forested, the island, but when humans came there, the forests were destroyed, and it's now this sort of barren island. But there's been a lot of discussion about the Rapa Nui because several anthropologists have claimed that there's traces maybe of Native Americans on this Polynesian island. It's the most eastern one, most close to the Americas. And there've been all kind of evidence arguing that, in fact, there's been some interchange between Native Americans and these Polynesians in these Pacific islands. Let me see if I have, I'm missing a slide. Oh, there's a slide. Okay, good. All right. So what we know about the Polynesian islands is that they were colonized within the last 3,000 years, roughly. And using a route, something like this, where Rapa Nui or Easter Island is all the way out here and has been colonized about 900 years, 80 in year 900, so about 1100 years ago. That's where the Polynesians came to Eastern Island. The colonization started, we think, in or around Taiwan. And then people developed boating technologies, moved down here. Then they admixed with people from New Guinea, and the Polynesians we have today are admixed between people from Taiwan and people from New Guinea. But if you look at various, there's various cultural evidence that there might have been this exchange with South America somehow. And in part, there's certain words for certain crops that are used both in South America and in the Polynesian Islands that are the same. So that's odd. Why would you have the same words for that? That is pre-colonial, from before there were Europeans there. So it seems like there might have been contact before there were Europeans. So we did here, this is about 7 years ago, a project with this. And what we had was data from Rapa Nui, people from the Eastern Islands, from a bunch of other Polynesian islands, Native Americans from South America, and then some Europeans. And then the first thing we did was we made an admixture plot. And this admixture plots you get, if you use these populations, you get Europeans largely unadmixed, Native Americans unadmixed, Polynesians as unadmixed, except these Rapa Nui that we're getting. And what you can see here is that they have some European ancestry, and they have some European that's blue, and they have some Native American ancestry in red. But what you might notice already now is that there's a lot of variance in the European ancestry among individuals, and much less variance in the ancestry that they have from Native Americans. So that is already suggesting to us that maybe the ancestry that Rapa Nui have, that is Native American, did not come in just with Europeans. That the European ancestry came in later. There's a high variance here. And the Native American ancestry is older, because there's less variance among individuals. All right, so we made models of this where we made different models of the admixture. Model 1, in which you allow the admixture from Native Americans into the Rapa Nui to be older, could also be younger. It was also allowed to be older than the admixture for Europeans. So you have two different admixture times, T European and T Native Americans. And then we have another model where they have the same admixture time, so that the European and the Native American ancestry came in at the same time. Okay, why are we so interested in this? Why is that the question? The question is, again, if this ancestry came into the Rapa Nui with the Europeans, with colonialists that came, or does it predate that? Because if it predates that, that shows there was contact between the Polynesians and the Native Americans. Okay, and then we fit models of tracts, of ancestry tracts. We use a methods, one of the established methods for estimating ancestry tracts, and then we fit those, and this is just using some simulation models. Fit with M1 and M2. And remember, M1 allows different admixture, and M2 does not allow different admixture times from Europeans and Native Americans. And what you can maybe see here is that we have a hard time fitting M2 doesn't fit so well as M1. And if we go back here, oh, I don't have it, but the maximum likelihood estimates that we get is 21 generations here for the Native American and 4 generations for the European ancestry. And we get a much higher likelihood for the M1 model. The M1 model is supported, and with strong evidence that this is about 600 years ago, and this is about 120 years ago, that the European admixture is about 120 years ago, but the Native American is about 600 years ago. And this European admixture 120 years ago, that's coincides well with when Europeans started to actually settle on the island. And this, the 600 generations, is of course pre-colonial. So that would be a mixture that happened before there were any Europeans there. And there actually been a recent paper that was published in Science where they have data from all over the Polynesian island, and where they show the same thing, where they also show using basically this same kind of method, that the tract lengths are such that that you can only really fit the data by assuming there was Polynesian Native American admixture that happened before Europeans got there. And there's another island than just Rapa Nui, than just East Island, for which it's also true. So there seems to have been Native American and Polynesian contact before Europeans. And we see the genetic evidence of that. Okay, one more example here that I'll entertain you with. Is there something in the chat? Is that a question?
I don't know if you meant this paper. Yeah, but this one is in Nature from last year about Native American gene flow into Polynesia.
Yes. Yeah, so that was this, yeah, that's paper. Thank you. I can't see the chat when I'm sharing my screen. That's why. So I can't see what's in the chat. So if anybody puts anything in the chat, just speak up. So there's a paper that basically showed the same thing and confirmed now, in fact, there is this kind of admixture between Native Americans and Polynesians. All right, any questions about that before I go on to another example? Okay, so this is another example now that has to do with the Neanderthal admixture. And perhaps of all of these admixture stories, what that's been most interested in is trying to understand how the Neanderthal admixture really worked, where it happened, when it happened, and so on. And one thing that's helped a lot with that is that you have Neanderthal admixture also in ancient humans that are old. And this is a project we also did some years ago from an individual that's about 37,000 years old, the Kostenki individual. So this ancient DNA, a relatively high, good quality genome from an individual that died 37,000 years ago, in Russia, Kostenki, and we sequenced the ancient genome of this individual. And one of the things we then looked at was admixture tracks of Neanderthal DNA in this individual. So this plot is a plot of admixture tracks in different individuals. These are a bunch of modern individuals, and these are ancient individuals, where the oldest one is Kostenki, that's here. Malta here is about 25,000 years I believe. Kostenki here is, 36, 37,000 years old. And these are the track lengths. And what you can see is that the track lengths are much longer in Kostenki than in the modern individuals. And Malta has the second longest, and it goes with what you would expect. The old individuals have the longer tracks because there have been fewer generations with recombination in those individuals to break up the tracks. So based on that, we can now take the tracks and we can estimate what is the admixture time between human and Neanderthals. And if you assume there was only one admixture event happening in one generation, we know that's not true, but as an approximation, we get here that's about 54,000 years old based on Kostenki. And it gives much better estimate. Use this old individual that's much closer to the time where admixture happened. You can get better estimates than if you use modern individuals. And it fits sort of relatively well with this, admixture tract length distribution here and here, given what we know about recombination in the human genome. So that's nice to see. It's sort of a validation that, we have these models about how these tracks should be broken up through time. We in fact see that when we look at a Neanderthal admixture in humans and take ancient humans and modern humans, we actually see kind of what we would expect, more or less. And the reason why I say more or less is that there is a tendency, has been argued, for less Neanderthal ancestry through time. Now, this has been challenged, and there's some debate about, is that true? Actually, that's a negative correlation here, because there might be actually some inference errors in the method that's been used, and essentially and ignores some genetic drift. It uses some reference populations and ignores genetic drift in those reference populations, and that can create some biases. But there's still some evidence that there might be some declining Neanderthal ancestry through time. Now, why would that be? Well, people argue selection, but perhaps the more direct evidence that the selection against Neanderthal ancestry in humans is that there's a significant paucity, so a deficit of Neanderthal tracks around protein coding variants. So if we look in the part of the genome that is functionally important, there's less Neanderthal DNA in those parts of the genome. And that's really hard to explain why that would be without selection. So there's broad consensus that there has been selection against Neanderthal DNA in the human genome. Now, why is that? Perhaps the most popular theory for that is that it's called what evolutionary biologists call Dubchansky-Müller incompatibilities. And to explain what that is, I'll have to talk a little bit about speciation and how species are formed and so on. That's where they arise. So imagine that, and that's where there's a lot of theory about these Dubchansky-Müller incompatibilities. And the question that this comes from is, how could you, when you see different species, you have genetic incompatibilities, but how could they possibly arise? And the problem is, if you have a mutation that creates an incompatibility, that mutation should always be selected against. So how can you accumulate incompatibilities between two species, since anything that creates an incompatibility must be selected against? And the solution that Dubchansky and Müller came up with was that it has to do with interactions between the two mutations. So imagine you have an ancestral genotype here, two loci, the A locus and the B locus. Then these two, they split up from each other into two species, two isolated populations, and mutations can now start to accumulate. In one species, you have the little a mutation here, and it's benign in the context of the capital B allele. In the other population, you have a little b mutation occurring, and it's also benign. It has no negative effect in the context of the capital A allele. So eventually, by genetic drift, or maybe they're in favored, and there could be selection, you have a haplotype here that's capital A, lowercase b in one species, and lowercase a, capital B in the other species. Now, when you make it a hybrid, the hybrids could have reduced fitness if there's an interaction between the two. That is, it's not good to have little a if you also have little b. Why might that be? That might be two proteins that have to bind each other. A codes for one protein, B codes for the other protein. And they can bind as well enough if lowercase a and capital B. They can bind well enough with capital A and lowercase b, but they can't bind well with lowercase a and lowercase b. Then they don't bind anymore. So lots of cases, biological cases, where we have cases like that. So there's an interaction, there's epistasis here. In that case, you can accumulate mutations that makes the two genomes incompatible. So the hybrid has reduced fitness, maybe inviable, or it is infertile, as we see in many, if you cross many wild species, you will find that. So you can accumulate these genetic incompatibilities in that way. And what many people think is what we see when we see selection against Neanderthal DNA in humans, it's because of these kind of incompatibilities. And since there was less Neanderthal DNA coming into humans, it's the Neanderthal DNA that's been selected against. If humans integrate into Neanderthals, it will be the human DNA that has been selection has been working against. So that's one hypothesis. Another hypothesis is that it's simply because Neanderthals had a lower effective population size. Okay, so this is a model other people have been estimating of a simple model of the history of human Neanderthals. Human populations that went through a bottleneck at some point and then expanded in size. To make a realistic model, you have a much larger size expansion, but this actually models much of what happened until a few thousand years ago and can model most human variation just fine with a model like this. About 18,000 generations ago, humans and Neanderthals were split up for 16,000 generations, and then 2,000 generations ago, there was admixture from Neanderthals into humans. An important point is that the Neanderthals have very little genetic diversity. They would have had a much smaller effective population size than humans. Because of that, they might have accumulated more deleterious mutations. So if you have a small effective population size, selection is not very efficient. And that means that slightly deleterious mutations, they are much more likely to go to fixation. So small populations accumulate deleterious mutations. And that's one of the reasons why that small populations might go extinct, for example, because they keep accumulating deleterious mutations. They don't have a large enough population size for selection to act efficiently. They can't, in one way to think about it, they can't every generation kill off enough individual to get rid of all of the deleterious mutations. So you have an accumulation of deleterious mutation. And if we look at, there's a way to predict whether a mutation is deleterious or not. And if we look at the Neanderthals and try to predict that, we can actually show that Neanderthals actually have more deleterious mutations predicted than humans do. So there's all kind of evidence that Neanderthals actually had more deleterious mutations. Now, what we did in some modeling, and that's Kelly Harris that did this work, my former postdoc, who's now a professor at the University of Washington. We simulated under a distribution of fitness effects of mutations that other people estimated. This model of human Neanderthal history, very simplified, but can generate most of the patterns we see in the data, simulated under that model with this distribution of fitness effects. So then what we get if we do that, and is that under this model, the relative fitness of Neanderthals to humans is like this. There's about, because smaller population size of Neanderthals, they have reduction of fitness about 25%. They have much lower fitness than humans. And again, what we're doing is we're not modeling that in itself, that we're not making the assumption that they have more deleterious mutations or anything like that. It's simply just a consequence of using the same distribution of fitness effects of new mutation in these populations, and Neanderthals have a small effective population size. So they end up with more deleterious mutations and have lower fitness. And then when the populations come together, then what will happen is that there will be a reduction selection against Neanderthal DNA. And so if I go back to this slide, at this time, they come together, Neanderthal DNA goes into the human population. But because of all the deleterious mutation in the Neanderthals, the Neanderthal DNA will be selected against. And what we see here is what we get, the Neanderthal ancestry fraction as a distance to a linked locus with a deleterious allele. After 20 generations, 800 generations, 2000 generations, and compared to some theory that we developed. And what you can see is that you get this pattern where you get less Neanderthal DNA close to the functional regions where you would have the deleterious mutations. We can plot here the Neanderthal admixture fraction as a function of time. And this is what you would predict from this model, that it should go down like this. And that model very well describes this pattern that you see in the genome of not enough Neanderthal DNA about the protein coding regions. That's exactly what you tend to see. But there's another interesting thing about this worth noticing is that to get, if there's been this amount of selection, and that would, I think, go for both our model and the Dobzhansky-Müller incompatibility model, to get the amount of Neanderthal admixture as we see today, the initial admixture should have been 10%. Otherwise, we can't explain the pattern that we see in the data of evidence of selection. So in fact, when initial admixture happened between humans and Neanderthals, it was 9-1 or 10% admixture. And that's interesting because it matches these population sizes that people have estimated of the effective population sizes. Okay, so a model that fits this perfectly is if at this time where there's admixture, they simply merge together these populations. It's not like the Neanderthal went extinct. It's just you take human Neanderthals and you just merge them together. They start interbreeding freely. And that actually predicts everything we see in the data. That the pattern that we see of genetic variability in humans, of Neanderthal in humans, entirely compatible with that model. That's what you would predict. Okay, so here's, it depends on things, on dominance of the mutations and various assumptions, you will get different things. But in some sense, what this predicts is that the Neanderthals did not go extinct. They just simply merged together with humans. But because there are many more deleterious mutations in them, then the DNA from the Neanderthals has been weeded out, has been removed by natural selection. So there's only 3% of the Neanderthals left today, 3 to 40% in modern humans. All right, so I talked a lot about, let me hear, are there any questions about this? Anyway, for people that are interested in these questions, there's lots of things to do to try to explore the patterns that we see of Neanderthal DNA and also Denisovan DNA in different human populations, its spatial distribution and its distribution among individuals. To really try to get at how did this happen? Not only, making an estimate that that mixture happened 54,000 years ago, but in which time periods did it happen? Were there bottlenecks after it happened? What is the demographic history of Neanderthal and Denisovan integration into humans? There's lots of data to work on that. There are lots of questions on that. And I think we're starting to have a mathematical framework and methods where we can answer these questions, but they're not that much work. There's some, but not that much. I'm really doing detailed work trying to answer these questions. So people that are interested in more biological problems, there are lots of problems taking these kind of methods and really going in detail, trying to get at the history of Neanderthal and Denisovan admixture. That's not really fully explored. So if some of the students out there looking for projects, I think there's some that are more on the data analysis side of things, but also include some theory, then I think that there are some good projects out there with that. All right, so I talk mostly about humans. But you actually see the same thing in many other species. On almost every species you look at, there's extensive admixture between things that we previously thought were species. We say humans and Neanderthal, they're anatomically very different from each other, so we define them as different species. Here's some that we definitely define as different species: the yak versus the bison versus the bison. That's the European bison that you find, in Europe, still in the border between Poland and Belarus, I believe, and maybe also a few other places it has spread to. The Bangtan, the Gayal, the Zebu, and then, the normal cattle here. Anatomically very, very, very different. But if you analyze their genomes, you'll find all this evidence of integration between them that is, quite easy to find, where you find, for example, the yak and the cattle. There's been, after humans brought cattle up to Tibet, they have integrated. There's been integration between yak and cattle, but also between several of the other species, wild species that are very different from each other, where there's been integration between them. Here's another example from the pig, Sus scrofa. That's the pig. That's also the domesticated pig. And then we worked on a project on looking at genomics, particularly on sex chromosomes in Sus scrofa. And this is one of these plots where each column again is the SNP and each line is a haplotype. And this is the X chromosome. And what you can see is that this is massively differentiated haplotype between northern Chinese wild boars, European domesticated pig, and European wild boars. And then these southern Chinese domesticated pigs and southern Chinese wild boars. And then the northern Chinese domesticated pigs and wild boars, they seem to be somehow recombinant between these two. So somehow there's recombination suppression here. You would expect it to be inversion. My collaborators tell me there's no evidence this should be, I don't think it's an inversion, but there's definitely recombination suppression. I still think it could be an inversion. That just hasn't been discovered to be an inversion. And then there's been recombination here between just one recombination event. But the interesting thing is that when we make phylogenies of this, we get a really extreme pattern. So this is what you see in most of the genome. You have here the Chinese wild boar, European wild boar, European domesticated pig, the Chinese domesticated pigs, Southern Chinese wild boar, Sumatran wild boar. These here are also scrofa. And then you get some other species out here, some outgroups, some quite divergent outgroup. So that's what you see in the entire genome, except in this region, what you see is that the Southern Chinese domesticated pig, Shan wild boar, and Sumatran wild boar, they group with these ones. But the European goes out as an outgroup, and it's really highly, highly divergent, really, really different. So it suggests that there's some really old integration event that goes between, something that's really outside the genus, highly, highly divergent, that's come into the European boar. And what that is, we don't know. We can't find another species that match this DNA. It's highly divergent DNA. There's no other species that matches it. So we don't really know what it is, but it could be something that happened millions of years ago from a species that's now extinct. There have been many, many other pig-like species in Eurasia. So there's a lot of different things it could be that now doesn't exist anymore. And the reason why we see it is because there's this recombination suppression. And then we see these long tracks here because there's no recombination here. But if there was recombination, we wouldn't see it. So likely what we think is things like this kind of divergent admixture or integration between different species is probably quite common. Probably see it a lot. It just, usually because the tracks are being blown up to becoming so small, we don't notice it anymore. But because of this having to have recombination suppression, we actually see it. So this is the spatial pattern of where you have these different haplotypes in China. So this is China, where you have Tibet over here. And this is southern China, where you have one haplotype, and there's this other haplotype in northern China. All right, so that's that. We think integration and admixture has been really important in human evolution because it's been spreading. We see many genes in human evolution where they are really important functionally for adapting to local environment, and we got them from other hominins. We see integration between species all the time, often in functionally really important regions. And we also now have a mathematical framework, I think, for really getting at these admixture tracks and understanding them. So I think lots of interesting research to be done on really taking the theory and connecting that to a lot of this data that's out there and trying to understand admixture better. And these are some of the people that have been working on various parts of this research I've been talking about today. So I just want to mention some of them. Mason Liang, who is who graduated and then went out to the tech industry to make a lot of money instead of doing basic research. And so he's not doing this kind of research anymore, unfortunately. Fernando Racimo has been working on some of these models. He's now professor at the University of Copenhagen. John Poole, who's now a professor at University of Wisconsin-Madison, Matteo Fumagalli, who became a professor now at Imperial College, Kelly Harris, who's now a professor at University of Washington, and Emilia Huerta Sanchez, who became a professor at Brown, where she is now. And these were all trainees in my group who worked on these different research. And of course, lots of the data, lots of people that generated data that we've been talking about. All right. So that's all I have for you, had prepared for you today. Are there any questions about anything?
I guess not anymore.
No questions? Maybe what I will do for the next lecture is to make it a little interactive. I speak for 2 hours here. I'm all, so on my throat from speaking for so long time. And you guys probably just sit and fall asleep as I keep talking and talking. I will prepare some questions for you next time. So I think I will stop myself talking and then have some questions for you that I'll ask you to discuss. So it's a little bit more interactive.
That would be just great.
Yeah, because if you don't interrupt me, then I just go on and go on and go on. And I think we'll do it slightly different on next time. So next time, what I will try to talk a little bit about is more on inference methods using stochastic simulation and population genetics. So next time I will talk about microchain Monte Carlo methods and important sampling methods for estimating population genetic parameters. So computational intensive stochastic methods for parameter estimation. And again, for all of this, when I talk about it, I assume that you already know the material from the lectures from the previous year. So I'm not repeating those lectures because Vladimir told me that it's many of the same people and you had access to those videos of that. So I'll also assume that for my next lecture. All right, any questions about anything?
No.
All right, so I will end the lecture here now today, and then I'll try to do it a little more interactive on Thursday when we meet again. All right, thanks for listening in today, everybody.
Thanks.
Thank you.
Bye-bye.
Goodbye.